Hello everyone and welcome to Instant Biology by Dr. Neelap, and the topic that I have to discuss today is DNA replication in prokaryotes. And in this video I would be concerned with the initiation process. That means you would be knowing that replication can be divided into initiation, elongation and termination. In this video I would be focusing upon initiation process, and as you can see that I am using English language, so this video would be in English. I have already made a video in Hindi, so if you are comfortable in Hindi you can switch over to the Hindi version. So in this video, as I said, that we would be talking about the replication, and in replication also prokaryotic replication, and specifically talking about initiation. So whenever we are talking about prokaryotes, then we focus or we use a model organism, that is E. coli. E. coli, as you would be knowing, that it is having circular DNA. Okay. In case of eukaryotes, the DNA is linear, but when we are talking about prokaryotes, the DNA is circular. What does circular mean? So it is starting from one point and then it is moving and again comes to just the point opposite to the initial point or the starting point. Okay, and this DNA, that is circular DNA of prokaryotes, is double stranded in nature. Next important point is that the replication in prokaryotes, or to be more precise in E. coli, it is bidirectional. What does bidirectional mean? So as you would be seeing that this is suppose the circular DNA. Okay, so this is suppose the circular DNA. So this circular DNA will undergo a bidirectional replication. That means the replication will start from here till here, this way, and also from here till here, like this way. That means it will be bidirectional. That means starting from one point, it will approach like this. Okay, that is why I am saying bidirectional. One is this direction and one is this direction. Okay, and the point from which the replication starts, the point at which the replication is starting, that point is called as origin of replication. Now in case of prokaryotes, there is only one origin of replication. That means the replication will have to start from one single point in case of prokaryotes. For example, if I show you over here in this figure, then you would be able to understand that suppose the replication in this case is starting from this particular point and it is moving in this direction, and it will also move in this direction. Okay, so in this photograph also, this book photograph also, you can see that the replication is taking place in both the directions. For example, you can see starting from this side, it is taking place in this direction also and in this direction also. Okay, so this is called as bidirectional replication. One more important feature is that because in or during this replication in prokaryotes, a symbol theta is displayed in the process of replication. So you might be knowing that this is the symbol of theta, and this theta is displayed during the phase of replication. Over here you can see that this theta is displayed. That is why the prokaryotic replication is also called as the chromosomal replication. I am talking about. In prokaryotes, plasmids are also present, but the plasmid replication is called as sigma replication. But when we are concerned with the chromosomal replication, it will be called as theta replication, because it is displaying a theta, that means the shape theta, during the course of replication. So what we have discussed up till now, that there is bidirectional replication. The prokaryotes have circular DNA and it is double stranded in nature, plus the theta symbol is displayed. That is why it is called as theta replication. Now this is the method, or this is a GIF through which you can see that replication is starting from one particular point, and then both the newly formed strand is separated from the older strand. This is what you can see over here. Okay, so this is a GIF of how replication is done. Okay, so now let us talk about the proteins that are involved in the replication. Since you already know that we are interested in this particular video to talk about the initiation phase of replication, so we would be talking about the proteins that are involved in the initiation process. The first protein that is involved in the initiation process is DNA-A. Now what does DNA-A do? Now DNA-A actually is a protein that binds to the DNA origin of replication. Now let us understand this in detail. Actually the E. coli origin of replication is around 250 base pair in size. You can see that the E. coli origin of replication region is around 250 base pair, and this origin of replication is having two types of areas or two types of regions. One region is one region is having 13 mer repeats and the other region is having 9 mer repeats. Now what does this mer means? Mer means actually nucleotides. That means there is 13 nucleotide repeat and there are 9 nucleotide repeats. So this happens in the origin of replication only. Okay, so origin of replication is a place where 13 base pair repeats are present and 9 base pair repeats are present. Okay, now in the figure, in the photograph, you can see that 13 mer repeats can have G, A, T, C, T, N, T, 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 A, T, T, T. So this is one example of the 13 mer repeat. Similarly, there are other examples also, and this is about the 9 mer repeats. There are multiple examples of the 9 mer repeats also. Now these two 13 mer repeats and 9 mer repeats are present in the origin of replication. Now what does DNA-A do? DNA-A binds with the 9 mer repeat that is present in the origin of replication. Basically it is identifying the origin of replication. So if someone asks you what is the purpose of DNA-A, so DNA-A is just identifying the origin of replication through the 9 mer repeats. It binds to the 9 mer repeats. But more importantly, it is having two binding sites. Okay, so DNA-A is having two binding sites, two DNA binding sites, and through one it is binding to the 9 mer repeats. Whereas I would be talking about the other DNA binding domain or DNA binding site later on. Now coming to the next protein that is involved, it is single-stranded binding protein. Now what is the use, or what does this single-stranded binding protein do? Okay, let me just show it to you through an example. Now suppose this is my double-stranded DNA. Okay, I should have shown the circular DNA, but let us consider this as a circular DNA, and this is double-stranded in nature. Now you know that for the purpose of replication, both of these strands should be separated out. One strand is this and the other strand is this, and they need to be separated. This separation is done by helicase. You already know this. Now, but even after separation, the complementary bases are present. Complementary bases are the same. That means suppose A is present over here, T would be present over here, C is present over here, G would be present over here. That means complementary bases would definitely be present. And because complementary bases are present, therefore they can again bind with each other. They can again have the hydrogen binding or hydrogen bonding. We would not want that, because the helicase is requiring ATP for breaking the hydrogen bonds. If again the hydrogen bonds form, then it would be wastage of the ATP. We would not want that. Therefore, on both of these strands, certain proteins bind, and the name of these proteins is single-stranded binding proteins. And what does the single-stranded binding protein do? It does not allow realignment of the open strand. Okay, both strands are open, so it will not allow the realignment, or it will not allow the re-annealing. Re-annealing would not be allowed. This is the purpose of single-stranded binding protein. So this is what I have written. You can see this, that it stops the subsequent re-annealing of the separated strands. Next, let us come to DNA-B. DNA-B is actually helicase, and helicase is a hexamer. That means there are 6 subunits present, and it is called as DNA helicase, since it is breaking the double-stranded DNA into single-stranded DNA. It is just opening up the double-stranded helix. Next, let us come to DNA-C. What is this DNA-C? DNA-C is called as helicase loader. Now, what do you understand by helicase loader? I have already written that it is involved in opening and placement of the DNA helicase protein ring and placement around the single-stranded DNA. So, actually understand this, that helicase, just let me show you, that helicase cannot by itself go and bind on this DNA strand. It cannot do that. So it will require a helicase loader. What will this helicase loader do? It will open this it will suppose the helicase is just like this. It is a protein like this, and this helicase can open and it can close like this. Okay, it can open and close like this. So why do you need to open it? We need to open it so that it can bind to the DNA like this, and then it closes, and suppose it needs to get out of this DNA, it opens and comes out. But it cannot do this by its own. I mean, the opening of the helicase ring does not happen or does not occur by its own. It needs to be done by another protein that is called as DNA-C. And what is this DNA-C? DNA-C is also called as helicase loader. It is involved in the opening and placement of this DNA helicase around the DNA strand. Okay, so placement around the DNA strand, this is the function. Now let us talk about the DNA-G. Next protein is DNA-G. Now what does DNA-G do? Now DNA-G is primase. Okay, so it is primase. Now what do you understand by primase? So primase is an enzyme that would synthesize primer. Now you would be knowing that primer are short oligos that are required by the DNA polymerase for extension of the DNA strand. That means, suppose this is my DNA strand, this is my DNA strand. If I want to copy this DNA strand, I want to use this as a template, I will have to synthesize a primer over here first of all, and this primer cannot be synthesized by DNA polymerase. So this primer needs to be synthesized by another enzyme, and this primer will provide a 3' OH end that will be extended by the DNA polymerase. So which enzyme is responsible for synthesizing this primer? It is called as DNA-G. This DNA-G is actually a RNA polymerase. That means it synthesizes RNA. That means the primer is having RNA nature, not DNA nature. Okay, I believe you would have understood that DNA-G is a primase. In this page we have understood about DNA-A. What does DNA-A do? It is required for identifying the 9-mer and 13-mer units, basically origin of replication. It is used to identify single-stranded binding site. Basically single-stranded binding protein actually prevents the re-annealing of the separated strands. DNA-B functions as a helicase, DNA-C functions as a helicase loader, and DNA-G is functioning as primase. Now let us move on to the next slide. Now you can see over here I have shown you all the proteins that are taking part in DNA replication. You can see that this is DNA-C. DNA-C is functioning as helicase loader, and you can see that it is helping in opening of the DNA helicase. DNA-B was actually your DNA helicase, and these are the other things that you do not need to focus upon. These are the 2 strands of DNA that are being opened. Okay, so this is basically a diagrammatic representation of the proteins that I did talk about. Now, proteins involved in the chain elongation and Okazaki fragments. So we are not going to talk about these proteins as of now. but since they are a part of the elongation process, and we would be making a separate video on the elongation. However, I'll just tell you that which proteins are involved in DNA elongation. So in DNA elongation, DNA polymerase, single stranded binding protein, DNA gyrase and DNA ligase are involved. We would talk about them in more detail in the upcoming videos. Now let us move on to the next slide. For the termination, there are other proteins that are required. Example is terminus binding protein, that is also called as TUS, and DNA topoisomerase, that is DNA topoisomerase 4. Okay, so these proteins are involved in DNA termination. Again, I'm not interested to talk about these proteins as of now, because in this video we are focusing upon initiation only. Let us now move towards the next slide, and this slide focuses specifically on initiation process. Now, what happens in the initiation process? Now we will talk step by step. In step number 1, what happened is DNA A actually bound to the origin of replication. We have already talked about this. Where does it bind? It binds to the 9-mer region, 9-mer repeat region that is present on the origin of replication. So does DNA A require ATP for doing this? Yes, of course. It cannot do this without ATP, so it utilizes ATP. Next, melting of duplex DNA at OREC, 13-mer repeats. What does this mean? This means that 13-mer repeats, there would be 13-mer repeats on the origin of replication. So what happens on the 13-mer repeats? Remember, DNA A initially bound to the 9-mer repeats, and its impact was seen on the 13-mer repeats, and the 13-mer repeats, now the DNA actually opened. That means the hydrogen bond initially broke apart at the 13-mer repeats. Okay, so this is what is written over here: melting of duplex DNA. Melting means breaking of the hydrogen bonds at the 13-mer repeats. Now, as I initially said that DNA A had 2 binding domains, or has 2 binding sites, 2 DNA binding sites rather. So these 2 DNA binding sites, with these 2 DNA binding sites, it is binding to the 9-mer repeats and also to the 13-mer repeats simultaneously. Now, coming to the next point, separation of DNA and more than 20 base pair within the 13-mer repeats and open complexes formed. So that is what I just explained, that at the 13-mer repeat site, what happens is that 20 base pair, around 20 base pair, I will just move myself. Yeah. So at the 13 base pair repeat site, what happens is more than 20 base pair region is opened. Okay, that means open means that there is a open complex formation. That means the hydrogen bond between the 2 strands would be broken and the strand would now be open like this. It was initially closed like this, now it is open. I believe you would have understood. Now let us move on to the next portion. Just give me a minute. Yeah. So let us talk about step number 2. Step number 2 is loading of the DNA helicase. Now I already told you that this DNA helicase, or this DNA B, you are also calling helicase as DNA B, isn't it? So DNA B or helicase has to clamp around the 2 single stranded DNAs. Do you remember at 13-mer repeats, the DNA has already moved apart, or it has already formed the open complex. Now DNA helicase just has to bind. Now suppose, just let me show you with the help of 2 pens. Yeah. So this is the position of the 13-mer repeats. That means initially these 2 strands were bound together, but now they are a little apart from each other. Okay. So this is the region of the 13-mer. This entire region is 13-mer region. Now what will happen? Helicase will bind to this strand also, and will bind to this strand also. Remember, helicase was hexameric. Okay. So it clamps around each of the 2 single stranded DNA. So these were the single stranded DNA. It will clamp around this. It will also clamp around this. Okay, in the open complex. Next, DNA C also comes over here, and it helps the DNA B in order to clamp, in order to form the open in order to open the ring of the DNA helicase and close the ring and clamp around the DNA strands. So DNA C is helping it in doing this. Again, DNA B cannot do this without ATP. So ATP requirement would be there. ATP hydrolysis would take place in this process also. Okay, next, talking about SSB, you already know that SSB, what will it do? It functions to stop the reannealing. So SSB will do its function. Next, SSB are tetrameric in nature and they show cooperative binding. Why? What do you understand by cooperative binding? Cooperative binding simply means, now since they are multiple subunits, right? They are tetrameric in tetrameric in nature. That means there are 4 subunits. That means if one subunit is binding to the DNA, it will promote the other subunit also to bind with the DNA, the other subunit also to bind with the DNA. That means there is cooperativity in this. So, and this is sequence independent in nature. What do you understand by sequence independent? The single stranded binding protein can bind to any DNA strand irrespective of the nucleotides present on the DNA strand. This is what I wanted to say. Okay, now coming to this photograph, you can see that this is the 9-mer region, this is the 13-mer region, and what happens in the 13-mer region, you can see over here that the 13-mer region, basically it forms a open complex. Okay, so you can see this is the open complex over here, and the primer is also present where the primer formation takes place. And do you know what region or what protein or what enzyme forms the primer? DNA G. So DNA B is present over here, DNA C is present over here, ATP is also used. This is basically the entire thing. Let us now move forward and discuss about the next slide, and this slide is the DNA polymerase cannot initiate a new DNA strand synthesis. Understand this, that DNA polymerase does not have the capacity to start the DNA synthesis from scratch. That means it can synthesize DNA, but it cannot initiate the synthesis of the DNA. What does this mean? So in order to synthesize DNA, it has or it needs a 3' OH end. A free 3' OH end is a necessity. Now, for example, if I show you in this diagram, you can see that this is the single stranded template. Now template means this DNA strand needs to be copied. Now once the primer is attached, now this is suppose the primer. Once the primer is attached to the template, what does it do? It provides the free 3' OH. Now this free 3' OH is used by the DNA polymerase to bind the next dNTP over here. And what will happen if the free 3' OH is not present? For example, this is the template, and DNA polymerase is asked to synthesize the DNA strand right from the beginning. It won't be able to do because there is no 3' OH. So there is no 3' OH, it won't be able to do. That is why DNA G functions as a primase and it synthesizes the primer first. Okay. Now let me show you that how the strand is synthesized, or let us leave this up to the elongation phase because we are not interested in the DNA elongation as of now. I'm just interested in the working of the primase or DNA G to synthesize a primer. So over here a primer is synthesized, and we would continue about the chain elongation in the elongation video. Okay, so understand up till here that DNA polymerase cannot synthesize the DNA from scratch, so it will need the help of DNA G. Okay, now continuing, this is the step number 3. So step number 3, in step number 3, helicase will open the helix, it will bind to the primase. Remember, what did the primase do? It was making the primer, it binds to the primase to form the primosome. Okay, so primosome is formed. Next, therefore DNA primase or this DNA G synthesizes primer. Okay, So this DNA primase starts synthesizing the primer. What is a primer? It is a very short RNA molecule. Remember, why am I saying it as an RNA molecule? Because it is a RNA polymerase. That means this DNA G is nothing but RNA polymerase. And if there is RNA polymerase, it will definitely synthesize RNA only. So RNA is synthesized and the length of the RNA is less than 15 nucleotides. Let us come to step number 4. In case of step number 4, we are talking about the synthesis of RNA primer. So RNA primer has already been synthesized and primase was involved in synthesizing. Primase is RNA polymerase. I've also told you about this. And this primase, it is binding to both the strands of DNA. It is binding so that the primer can be synthesized on both the strands. Why? DNA polymerase cannot synthesize DNA from scratch. Next, now RNA primer, what does it do? RNA primer results in the release of the DNA helicase loader. Now what does this mean? So initially, do you remember we said that, suppose I am talking about one strand. So this was one strand and helicase is bound to this strand and helicase loader is also present. Now suppose this is helicase loader and it helped the helicase to clamp around the strand. Now what is happening, that this primer is getting synthesized. So primer is getting synthesized and it is pushing the helicase loader like this. Primer has got synthesized, it is pushing the helicase loader like this. So what will happen, the helicase loader will get removed. And also logically also you can understand that helicase loader is no longer needed. Why? Because helicase loader was only needed up till the helicase was not loaded or helicase was not clamped onto the DNA. But since now it has been clamped, helicase loader is no longer required. Okay. So helicase loader is no longer required. Then what will happen? So just a moment here. So it will cause the release of the helicase loader. So this will cause the release of the helicase loader, or in other words we can see that DNA C will get released. Next, primosome. What is this primosome? Initially I told you already that this is primosome. Primosome is actually DNA primase, that is DNA G, plus DNA helicase, that was DNA B. So DNA B plus DNA G is called as a primosome. Okay, so now let us talk about the initiation of DNA polymerization by DNA polymerase. So how does the initiation basically start? Look at this photograph. Now you can see I'm focusing upon this, the highlighted region. So you can see that this is a nitrogenous, or you can say that this is a sugar, and this is a DNA sugar, deoxyribose sugar. Okay, why am I saying that this is a deoxyribose sugar? Because in the deoxyribose sugar only 3' position has OH. Otherwise, if it was a ribose sugar, then this 2' position would also have a hydroxyl group. And this is again another sugar. This is nitrogenous base and this is the triphosphate. Okay, triphosphate. So you can see that this is a nucleotide, or DNTP, deoxynucleotide triphosphate. And this was also DNTP, deoxynucleotide triphosphate. It is having its OH, and this is the new DNTP that has come to bind. How does the binding take place? That is what I am telling you. The OH, actually it is having a lone pair of electrons. Okay, and you can see that this is the phosphate, and this phosphate is connected with oxygen over here, oxygen over here, oxygen over here, and oxygen over here. So what would be the situation of this phosphate? The situation would be that the electrons would be pulled from the phosphate in all directions, and because of that this phosphate would be slightly positive charged. And you know that oxygen, since it is having the lone pair of electrons, it will be negatively charged. So what will happen? This OH will attack, or this oxygen will attack on this phosphate and form the phosphodiester bond. Okay, so it will form the COPOC, phosphodiester bond. So in this way, phosphodiester bond will be formed. So phosphodiester bond means COPOC bond. Why am I saying COPOC? Because this is a carbon, this is an oxygen, this is a phosphate, this is an oxygen, and this is again a carbon. COPOC bond, it is called as phosphodiester. Why are we calling it as diester? Because COP bond is, it can be called as a phosphoester bond. Again this POC bond. So COP and this COP, these two bonds are present, that is why it is called as phosphodiester bond. This is the way in which each time new nucleotide will come, OH oxygen will attack on the phosphate and will form the phosphodiester bond. Again next time again DNTP comes and it will again do the same thing. So this will continue each and every time. Okay. So now in this particular diagram again you can see that this is the 13 base pair repeat, this is the 9 base pair repeat, DNA A is coming over here and this is causing the breakage or the opening of the double stranded DNA in the 13 base pair region. Okay, and then gradually the helicase gets activated and helicase starts moving in the different directions like this. Sorry, it moves like this in this and over here. This causes the opening of the DNA strand and soon the DNA G comes over here, that means the primase comes over here, primase synthesizes the primers, and following that the DNA polymerase comes and starts synthesizing the new DNA strand. So this is basically the set of events that take place. Next, let me just move on to the next slide. So it is said that the time required for E. coli to replicate the chromosome is 40 minutes, but the generation time is just 20 minutes. Okay, so this time is constant at 37 degrees centigrade. How come it is possible that the time required to replicate is 40 minutes whereas generation time, what do you understand by generation time? The time in which one bacteria will give rise to two bacteria. This is called as generation time. So how is it possible that in order the time required to replicate is 40 minutes and time required to divide is 20 minutes? The concept behind this is simply that on one circular DNA there are multiple replication forks that are continuously moving. So before one replication fork completes replication, there is just another replication fork that is just running behind it, and behind this replication fork another replication fork is present, behind this replication fork another replication fork is present. That means simultaneously many replication forks are running. It has been observed that approximately 6 replication forks are continuously running simultaneously on a DNA chain, on a DNA strand, on a circular DNA. I believe you would have understood this. Okay, so E. coli prevents reinitiation of replication immediately from ori C. Now this is an important point. What I am trying to say is that E. coli, it prevents reinitiation of replication immediately from origin of replication complex. That means suppose that one replication fork has completed, or one replication has completed from one DNA. One replication has completed from one DNA. So E. coli would not want that the next replication set starts straight away or immediately from the origin of replication. Why? Because there are chances, understand this, that there are chances that in the new strand that has been formed, there might be certain mutations that might have occurred. So we would want those mutations to be taken care of first. Okay. Next, so DAM methyl transferase, it methylates adenine within every 5' GATC sequence. Understand this. Suppose I am showing you that this was the DNA strand that was being copied. Okay. So from this DNA strand, another DNA strand has just been synthesized. So this blue pen or blue region is the new DNA strand that has been synthesized. But understand this, that from this black one, this blue one was synthesized, but there are chances that this might have some mutations. There are chances that the entire DNA strand over here was not, I mean to say, it was not just complementary present over here. There might be some mutations during the replication. So that is why E. coli does not want this strand to function as a template soon as it is synthesized. This is what is written over here. It does not want this strand, this new strand, to work straight away as a template, because it might be having certain mutations, certain abnormalities, and we need to take care about that first before using it as a template. Okay, I believe you would have understood this. So DAM methyl transferase, what is this? This is an enzyme, DAM methyl transferase. What does it do? It methylates adenine within every 5' GATC sequence. So wherever this sequence is present, 5' GATC sequence is present, what will it do? It will methylate the adenine. So it will do like this. That means on the adenine, a methyl group will be added. Next, at the time of replication initiation, genome is fully methylated. That means methylation on both the strands. That means when the replication starts, what is the condition? When the replication starts, what is the condition? When the replication starts, both these strands, the double-stranded DNA, when the replication is about to start and it is going to happen on this double-stranded DNA, both the double-stranded DNA, both the strands over here, they are having methylated adenine. That means wherever GATC sequence is present on this strand or GATC sequence is present on this strand, it will be methylated. Why? Because these are the old strands. They have already been scrutinized that there is no mutation over here. So at the adenine, methylation has already been performed. This shows that both the strands are safe. Okay, so that is what I am trying to say over here, that at the time of replication initiation, genome is fully methylated. Methylation on both the strands, because both the strands are safe. But just after replication, hemimethylated condition is present. Why? Because suppose this strand is replicated, and this new strand is this is the new strand. So this condition would be hemimethylated. Why this condition would be hemimethylated? This is the just after replication. Why this condition would be hemimethylated? Because the older strand was having the methyl group, but this is the new strand. This will not have the methyl group, because just after replication it will not have the methyl group. After some time, this new strand would also be methylated at the adenine of the GATC sequence. So this hemimethylation is detected by Sec-A protein. Okay, so this hemimethylation, that means a new strand and an old strand is bounded together. This is detected by the Sec-A protein. Now what does the Sec-A protein do? What is the purpose of the Sec-A protein? It reduces the rate at which 5' GATC 3' site undergoes methylation. So it is reducing the rate, it is slowing down the rate at which the methylation is happening on the adenine sequence. Next, it prevents the binding of DNA A to OREC. Also, it is not allowing the DNA A to bind at OREC. Again, the reason being the same, that we do not want, the E. coli does not want that the DNA replication starts just after the replication gets completed from the newer strand. It does not want to use this strand as a template just after the replication has completed, before addressing the mutations that might have happened over here. Okay, so next, it prevents the binding of DNA to OREC and stops the immediate reinitiation. Now, but this is very short-lived process. That means it cannot, this Sec-A cannot prevent the utilization of this particular strand as a template strand for replication. This cannot take place for a long time. So Fresh replication only starts before the completion of the previous rounds of replication, and as many as 6 replication forks may be active on a single chromosome. 6 replication forks may be active. So this is actually a multi-forked chromosome and also called as dichotomous replication. So there can be multiple replication forks. What was the replication fork? It was the region that was present like this. It was the region where there was an open complex. So this open complex regions can be multiple, almost 6 in a prokaryotic genome which is getting replicated. This type of replication is also called as dichotomous replication. Okay, so I believe this would have cleared your doubts about the DNA initiation, replication, and in replication initiation process we have completed. I believe you would have liked this video. If you have liked this video, do press the like button, do subscribe to my channel, and also share with your friends. One more important thing, that we have already started the GATE crash course. We have already started the GATE crash course. Okay, and in this GATE crash course we are completing all the important topics for GATE examination. If you are interested, you can join us. The contact detail is 8299134528. The batches that are running are in bilingual format. That means I am using English and Hindi both. And just a minute, I'll just write it correctly. So bilingual, the timing is from 5 p.m. to 6:30 p.m. and the cost of this course is rupees 3,800. We will be completing all important topics from all the units for GATE BT examinations. You would be getting the PDFs also, and you would be getting the recordings also. Again, this is a 1-month course. So if you are interested in joining this, you can contact us on this number. Okay, thank you so much and have a good day.